Today we're diving into one of the most transformative topics in modern biology, precise genome editing. Sounds technical, I know, but trust me, it's got real-life implications that are jaw-droppingly powerful.
Absolutely. I mean, we're talking about literally rewriting the genetic code that makes us who we are. And what's crazy is, it all started back in the 1950s with the discovery of DNA's double helix structure. That one breakthrough kicked off a whole cascade of technologies, right?
Right. Fast forward a few decades, and suddenly we're not just reading DNA, we're editing it. Think about tools like ZFNs, TALENs, and of course, CRISPR. Each one brought us closer to making precise changes in the genome. But let's back up a little. What makes gene editing precise anyway?
Great question. So, precise genome editing means you're not just cutting randomly, you're targeting exact DNA sequences and making specific changes. Imagine editing a sentence in a book. You're not tearing out the whole page, you're just correcting a single word. That's what tools like CRISPR-Cas9 allow scientists to do.
And it's not just about fixing typos. These changes can actually correct disease-causing mutations. There are over 3,000 known genetic mutations linked to diseases. So this tech isn't just cool, it's potentially life-saving.
Exactly. But here's the kicker. Doing this in real-life human cells is far more complicated than editing a Word document. You've got to get the editing tools into the cells, make sure they hit the right spot, and avoid unintended changes elsewhere. That's where challenges like delivery systems and off-target effects come in.
Yeah, delivery is a whole saga in itself. Scientists use everything from plasmids to lipid nanoparticles to viral vectors. AAV vectors, for example, are kind of like Uber for DNA. They deliver genome editing tools into cells, but they have a size limit, like a suitcase with a strict airline baggage policy.
Ha ha ha. Perfect analogy. And CRISPR's Cas9 protein is a bit of a heavyweight. It barely squeezes in. That's why newer, smaller variants like Cas12F are gaining attention. They pack a punch, but fit better in delivery systems.
Speaking of CRISPR, it's probably the most famous genome editing tool, right? What makes it so revolutionary?
It's the simplicity and flexibility. Traditional methods like ZFNs and TALENs need custom proteins for each target site, kind of like designing a new key for every lock. CRISPR uses RNA guides instead, so you're basically just swapping out a string of letters. It's modular, fast, and cheaper.
But it's not without issues. Off-target edits can still happen. Researchers have developed nickase versions of Cas9 and even fused it with other proteins like FALCI to reduce unintended cuts. Plus, engineering the guide RNA itself can dramatically improve specificity.
Then there's the newer wave of editing tools, base editors and prime editors. These don't even cut the DNA in the traditional sense. They act more like highlighters and pens, changing one letter at a time, or even writing in small edits without breaking the strand.
Which is huge, because DNA breaks can trigger repair mechanisms that sometimes do more harm than good. Imagine fixing a crack in a wall by sledgehammering it first. Not ideal.
Haha, yeah, not the DIY strategy I'd recommend. But seriously, these newer methods are game changers for treating diseases that involve single-point mutations, like sickle cell or certain types of inherited blindness.
And then we've got CAST systems, which use CRISPR combined with transposas to insert larger DNA segments. It's still early days, but it could be a future staple in gene therapy, especially for disorders requiring big genetic fixes.
Let's not forget the non-technical but equally critical piece, ethical and practical delivery. Getting these tools into human bodies safely is still a work in progress. Immune responses, off-target risks, and accessibility, all of that needs to be figured out.
True, and yet we've already seen early successes, like CRISPR being used to treat sickle cell disease ex vivo. Scientists edited a patient's stem cells and then reintroduced them. That's science fiction turned science fact.
So to wrap it up, precise genome editing isn't just a research buzzword. It's a rapidly advancing frontier with real potential to transform how we treat and maybe even cure genetic diseases.
Yeah, and while we're not quite at the designer baby stage, and hopefully never will be without serious ethical frameworks, the idea of editing our own DNA is no longer just theoretical. It's happening.
which leads to a big question for all of us to ponder. If we have the power to edit life itself, how do we choose to use it?
Would you like a follow-up script on the therapeutic applications, or maybe a deep dive into one editing tool like prime editing?